In this presentation, I will present our novel 3D RNA imaging technique for large intact tissue, such as adult mouse brain, which was published in Science last year. The video you are seeing shows 3D imaging of pulmonary messenger RNA in the brain of the 8-week-old mouse. Here is the outline of this presentation. I will introduce our RNA imaging method called TRISCO. First, I will explain the background of RNA imaging. Second, I will describe how we developed the TRISCO method, which consists of maintaining RNA integrity, controlling probe penetration, and clearing tissue for RNA detection. Third, I will show some application example of TRISCO method. Finally, I will share some practical tips for using TRISCO method. Let's begin with the introduction. RNA molecules play a central role in cell biology as essential intermediate in gene expression. They carry genetic information from DNA to direct protein synthesis, and they are widely used as indicators of cellular status. Because RNA is often expressed earlier than proteins, it serves as an early indicator of gene activity. Additionally, non-coding RNAs play diverse functional roles beyond protein coding. For these reasons, visualizing RNA is essential for understanding cell biology. Now let's consider the advantages of detecting RNA in 3D-dimensional tissue. Unlike proteins, RNAs can be detected using DNA probes, which is much smaller than antibodies. This enables faster tissue penetration. DNA probes are also highly customizable, allowing for detection of a wide range of RNA targets. Moreover, DNA probes are removable, enabling multi-round imaging of different targets in the same sample. Although RNA imaging in 2D section is well established, extending this approach to large intact 3D tissue has remained challenging, mainly due to technical limitations that haven't been fully addressed until now. When performing 3D RNA imaging in cleared tissue, we face several key challenges. The first one is maintaining RNA integrity. RNA is fragile and can be easily degraded due to the factors such as RNA contamination, high temperatures, or alkaline condition. So it's absolutely critical to keep RNA intact throughout both clearing and staining step. Second challenge is staining. To analyze entire tissue properly, staining reagents need to penetrate deeply. This becomes especially important when working with large samples like a mouse entire brain. Third one is tissue clearing. RNA detection must be compatible with tissue clearing protocols, and the signal need to be preserved in the cleared tissue. Additionally, the tissue must be sufficiently transparent for light sheet microscopy. We addressed all these challenges by developing a reliable method called TRISCO, which enables RNA staining in whole tissue, including large ones, such as adult mouse brain. First, let's talk about the RNA integrity. Why is it so important to maintain RNA integrity? This slide shows a typical example of how 3D RNA imaging can be affected by RNA contamination. Here, two samples are processed using the same TRISCO method. However, sample on the right side was incubated in PBS without proper precautions to prevent RNA contamination before hybridization. As you can see here, the signal was greatly reduced. This clearly demonstrates how critical RNA preservation is for successful 3D RNA imaging. Next, I'll guide you through the TRISCO pipeline and explain how we safeguard RNA integrity. The TRISCO pipeline consists of several steps: fixation, dehydration, permealization, bleaching, blocking, hybridization, signal amplification, trick processing, and organic solvent clearing. The protocol takes 15 days in total for processing a whole adult mouse brain. To protect RNA within the tissue, we adopted two main strategies. Firstly, we utilize polyvinyl sulfonic acid, a affordable chemical known to function as an RNase inhibitor. PBSA is included in the TRISCO pipeline during the in situ hybridization step and amplification step as shown here. Our research demonstrated that PBSA does not interfere with either hybridization or signal amplification. Second measure involves optimizing the protocol to minimize RNase activity. We start by fixing the tissue with paraformaldehyde solution, a condition in which RNase is inactive. And then we directly transfer the sample to methanol solution. This immediate transition is designed to prevent RNase activation under physiological conditions. After dehydration in methanol, the sample is directly transferred to formamide solution, which is known to protect RNA. After hybridization step here, the RNA forms RNA-DNA hybrid that is known to resistant to RNase, one of the major enzymes responsible for the RNA degradation. By implementing these protective measures, we've achieved a high level of reproducibility in our experiment. Next, let's delve into RNA staining. In the TRISCO pipeline, we use a method called in situ hybridization chain reaction or in situ HCR. Let me briefly explain how this technique works. In situ HCR is a novel in situ hybridization method that uses an initiator sequence and metastable hairpins. A pair of DNA probe, it's shown here, hybridize to the target RNA sequence and form the initiator sequence, as shown here. After hybridization, two metastable DNA hairpins begin to hybridize with each other on the initiator sequence without any enzymatic activity. That's why it's called hybridization chain reaction. This method is especially advantageous for 3D staining since the DNA probes and hairpins are small and can easily penetrate into tissue. However, achieving whole brain staining has remained a major challenge. Now, let me show you some typical examples of whole brain staining using the in situ HCR method. This image shows the result of the staining for Thy1 messenger RNA, which is ubiquitously expressed throughout the brain. Unfortunately, in conventional methods, staining often fails in the inner region of the brain due to limited reagent penetration. However, with our method, reagent can fully penetrate the brain tissue, as you can see here. Here is another example of messenger RNA staining. This image shows the staining of MALAT1, non-coding RNA that is ubiquitously expressed in the brain. In conventional methods, the signal appears only on the brain surface, as shown here. This may be because the DNA hairpins are consumed at the surface, preventing deeper penetrations. However, with our method, we achieve full penetration of the reagent and observe uniform signals throughout the entire brain. So what exactly makes our method work? The answer is simple. we adjust the temperature during in situ HCR. We found that by changing the incubation temperature, we can control the efficiency of the HCR reaction. Typically, in situ HCR is performed at room temperature, where the reaction efficiency is at its highest. But we discovered that reducing the temperature to 4 degrees decreased the reaction efficiency as shown here. When staining 3D samples, room temperature or 37 degrees consumed all DNA hairpins at the tissue surface. However, lowering the temperature to 4 degrees helps suppress reagent consumption at the tissue surface. This allows the reagent to diffuse deeper into tissue, as you can see here. By tuning the HCR efficiency in this way, we were able to achieve full penetration of the staining reagent throughout the entire frame. The final aspect of our method involves tissue clearing. In our approach, RNA is detected via DNA chains generated by in situ HCR method. It's crucial to ensure that these DNA chains remain in their original locations during the tissue clearing process. At the same time, tissue must be sufficiently cleared for light sheet microscopy. To meet both of these requirements, we developed a tissue clearing process called TRIC. It's here, which we apply before tissue clearing. Now, let me explain how we developed the TRIC process, how we managed to preserve the DNA chain while also achieving high tissue transparency. In terms of tissue clearing, we use the organic solvent-based method, which involves dehydration, delipidation, and refractive index matching. To preserve the signal during this process, we investigated how different buffers and dehydration conditions affect DNA chains hybridized in situ. This slide shows how much of the amplified DNA chain were retained under various buffer conditions. We tested methanol at different concentrations, PBS, 5-XSCT, and 2 different concentrations of TRISHCl buffer. We found that either 80% or 100% methanol or 500 millimolar TRISHCl buffer effectively preserved the DNA chains. In contrast, conditions like water, PBS, 20% methanol completely eliminated the signal, while others partially reduced the signal. Based on these results, we tried dehydrating the sample using high concentration of methanol after the in situ HCl process, and that led us to a new challenge. The challenge we faced was related to tissue transparency. We found that when brains were dehydrated with high concentration of methanol after in situ HCl, the sample did not become fully transparent. It's as shown here. In contrast, the standard iDisco plus clearing method, which starts with PBS followed by 20% methanol dehydration, produced well-cleared samples. This result suggests that the buffer component used in the in situ HCl protocol interfered with the tissue clearing. One might suggest using PBS after in situ HCl, but as I showed earlier, PBS failed to preserve the DNA chains. So we needed a buffer that would maintain the DNA chain without interfering with the clearing. Through research, we discovered that using 500 TRISHCl allowed the tissue to be both well cleared and retain strong signal. We named this TRISHCl wash step TRIC. Now I'll explain what actually happens during the trick process. The in situ HCR method uses SSC buffer, a saline sodium citrate buffer. We found that SSC precipitate during dehydration in methanol, and this precipitate cannot be cleared by organic solvent, making the tissue opaque in the clearing solution. This explains why the tissue didn't clear well, as we saw earlier. In TRIC, we washed the sample with trace buffer because it not only preserves the DNA chain, but also it dissolves during the methanol dehydration, thereby preventing precipitation. As a result, tissue becomes well cleared after tissue clearing. We name this technique TRIC, which stands for trace-mediated retention of in situ hybridization signal and clearing, and we refer to the full 3D RNA staining pipeline as TRISCO. Alright, now let me show you actual slice images of parvalbumin messenger RNA throughout the whole adult mouse brain. This is a horizontal section view, starting from dorsal side and moving toward the ventral. What I'd like to emphasize here is that the staining is highly uniform and the tissue is transparent enough to see through the entire mouse brain. Staining pattern matches well with the alien brain atlas. If you zoom in, you will see the staining reaches a single cell level, but I'll show you that in more detail later. Let's wait until the movie finishes. Here we show slice view examples of several inhibitory neuron markers, such as PBs, SST, VIP, NPY, GAD1, LHX6, as well as dopaminergic neurons, MAOA, DAT, AADC, and BMAT2. You can see the sparse distribution of cortical inhibitory neuron across the neocortex, and the localized expression of dopaminergic markers in the midbrain area, as shown here. These examples are part of our validated probe set. So far, we validated more than 30 target probes using our custom probe design script. Okay. I won't have time to show all of our data today, but I'd like to mention that we've already validated probes for a broad range of gene types using TRISCO. These include cell type markers, immediate early genes, and many others, all of which are highly relevant to neuroscience research. To conclude the method development part, I'd like to show you an example of multicolor imaging. We stain the whole brain with 4 colors: gray for nuclei, red for somatostatin, green for parvalbumin, and blue for GAD1. The movie on the left shows full brain view, and while the one on the right side provides a magnified view of the hippocampal area. As you can see here clearly, this image achieves single-cell resolution. Let's wait until the movie finishes. Here is a summary of TRISCO method. It's a robust and highly reproducible 3D RNA imaging method. Sample preparation takes 15 days in total for whole adult mouse brains. TRISCO is resistant to RNA degradation. Staining is fully uniform even in deep tissue areas, and method achieve high transparencies. We validated many different target genes, and the workflow supports multicolor imaging. Now I've explained the development of the TRISCO method. I'd like to show you some of its application examples. These examples highlight how robust and broadly applicable TRISCO is, both in neuroscience and beyond. Here is an application example in the mouse heart. The ventricle and atria were separately stained with TNNI3, which encodes troponin I3, shown in green, and NPPA, which encodes ANP peptide, shown in red. Here is an application example in the mouse kidney. Nephrine, shown in green, is mainly expressed in Bowman capsule. Here is an application example in the mouse lung. SCGB1A1, which encodes secret globulin, is mainly expressed in the small airway. Here is an application example in the mouse spinal cord. The staining pattern of CHAT, a cholinergic neuron marker, matched with the known expression pattern. We tested TRISCO in human brain as well. We stained the nucleus with DO1 and TBR2 mRNA, a marker of intermediate progenitor cells, in a human brain at 12.5 post-conceptional weeks. As you can see here, TBR2, shown in magenta, is mainly expressed in the inner subventricular zone. This example shows the application of TRISCO for analyzing brain activity following semaglutide treatment. Semaglutide is a GLP-1 agonist used for the treatment of type 2 diabetes and as a long-term anti-obesity medication. We compared the C-FOS protein, the left side is shown, and the C-FOS messenger RNA staining, right side, 2 hours after semaglutide administration. As you can see here, the activation site of the C-FOS, shown in red, are consistent across the whole brain in both protein and messenger RNA signal. This final application example demonstrates that TRISCO applied to tissue even larger than the mouse brain. Here, we applied TRISCO to the rat brain and guinea pig brain, which are approximately 4 times and 7 times larger than the mouse brain, respectively, in volume. As you can see here, we successfully stained TH in rat and SST in guinea pig. This example highlights the robustness of the TRISCO method in handling large tissue volume. Alright, in this final section, I'll walk you through the TRISCO protocol in detail and share some practical tips for successful implementation. One of the most frequently discussed topics at the beginning is probe design. Let me first show you the general strategy for probe design. Here is a schematic overview of how we design probes. This is your target RNA sequence. The first step is to perform blast search to find and eliminate any region with high similarity to other RNA transcripts. This step is important to ensure specificity. After removing these similar regions, we obtain targetable sequences as shown here. DNA probes are then designed to hybridize specifically to these unique sequences. This general strategy helps to maximize signal specificity and minimize background in situ HCR. Now you've seen the basic design concept. Let me explain how we practically prepare DNA probes. There are 2 main options. One is to order ready-made or custom-designed probes directly from molecular instrument. The other is to design the probe yourself. In our lab, we developed a simple probe design workflow using custom scripts available on GitHub. These scripts follow the same steps I described in the previous slide. For synthesis, we typically use IDT or pool to conveniently pull all probes into a single tube, though you can also order individual DNA probes and mix them yourself if you want to customize them. We provided the example for GAT1 and TPH2 in our GitHub repository. And of course, there are many excellent design tools from other groups as well. Feel free to use whatever best fits your needs. Regarding the hairpin, please make sure that the initiator type used in your probes matches the corresponding hairpin type. In our work, we use B1 to B5 hairpins, which are part of the third generation in situ HCR method published in 2018 and are purchased from Molecular Instruments. Next, I'd like to share how I usually perform probe validation experiment. For this, I recommend conducting validation using 2D section first. Here, the procedure we follow, it's starting from fixation, dehydration, cutting, formamide buffer hybridization, 1-day incubation of each probe on the hairpin, and you can observe section without clearing on confocal microscopy. The key point is sections are cut in 100% ethanol, a condition where RNase is inactivated. Then, just like the TRISCO protocol, we immediately transfer the section into formamide buffer. By following these steps, we can minimize the risk of RNA degradation and ensure reliable validation. Of course, if you are working RNase-free environment, you can use your standard method instead. Now, I'd like to walk you through the TRISCO protocol in detail. We are distributing a PDF file of the detailed TRISCO protocol, and in this presentation, I'll explain part of it. If you are interested in receiving it, please feel free to contact us. Okay, I'll show you the TRISCO protocol in detail starting from day 1. Here are the steps. Day 1, fix the tissue with paraformaldehyde overnight. Day 2, dehydrate the sample with methanol. Key tips here is to avoid washing the sample in PBS before methanol dehydration, as PBS washing carries a potential risk of RNA degradation. Day 3, treat the sample, delipidate the sample with dichloromethane, DCM. Day 4, bleach the sample with hydrogen peroxide. Day 5, methanol wash and hybridization. Here, you can pause the experiment in methanol or DCM for several days without affecting RNA detection. This flexibility can help with scheduling and workflow. Next, I'll walk you through the hybridization and amplification step of the TRISCO protocol. On day 5, we wash the sample in 50% formamide wash buffer and incubate it overnight in hybridization buffer. This is a blocking step. There are no need to rehydrate the sample with PBS beforehand. If your schedule is tight, you can leave the sample in formamide buffer for a few days. It won't affect RNA detection. Day 6, we begin hybridization. It's for 3 days. Day 9, we perform washes with 50% formamide wash buffer 3 times and 5-XSCT 3 times. Then we incubate the sample in amplification buffer overnight at 4 degrees. Day 10, we start amplification. It's for 3 days at 4 degrees. On day 13, we wash the sample in 5x SSCT. If needed, sample can stay in this buffer for a few days. Just keep in mind that the amplification should ideally be completed within 4 days to minimize background. After amplification, we move on to the trick step and tissue clearing in the TRISCO protocol. Day 13 is a TRIS HCl wash. Make sure to use only 500 millimolar TRIS HCl without PBSA since PBSA precipitate in methanol. Day 14, we wash sample with methanol to remove TRIS HCl and water. This step is essential for successful clearing. If needed, repeat the methanol wash to ensure complete removal. Day 15 is a DCM wash followed by DBE clearing. The sample should be transparent by the next day. Note that the signal will gradually fade over time, so it's best to complete imaging within 2 weeks. The final step is imaging with light sheet microscopes. I recommend the Braid system or ultramicroscope 2 from Miltenyi Biotec, as these systems are highly optimized for organic solvent cleared samples and extremely user-friendly. Here you can see a real scanning example of TRISCO cleared brain being imaged using Braid system. This scan is performed using light speed mode, a rapid imaging methods that enable high-resolution 3D acquisition of whole brain in approximately 10 minutes per channel at 1x magnification. For researchers working with large tissues such as rat brain, the Braid system is essential due to its speed and deep imaging capability. Acknowledgement. I'd like to express my sincere gratitude to our lab members and collaborators, especially Gubler in Denmark, the developmental tissue bank, and the big imaging facility. I also thank our funding agency for making this work possible. I'd like to thank Miltenyi Biotec for hosting this webinar and supporting the development of imaging technologies. Thank you very much for your attention.